All right, here's part 1 of Chapter 8, Microbial Metabolism, and I'm going to post a couple of short videos because this is a long chapter, but it depends on where your level is in understanding the material already to be dependent on how fast you work through this material. So you may not actually need to watch these. If you watch the other videos, the introductory videos, then you might, and then look at the PowerPoint notes, you might already have a good understanding of what's going on. So instead of taking lecture time to work through all of this, I'm going to go make a few short videos and then we can just work on assignments and practice problems during class. So Chapter 8 is microbial metabolism, the growth of the bacteria and what is going on the interior of the cell. And you should know already that metabolism is a chemical process. It occurs in every cell. It occurs in you. How much food can you eat without gaining weight? All right, that's your metabolism. Can you eat a lot of food, or do you really have to watch what you eat? But metabolism, by definition, are the chemical reactions that organisms use to break down substances to release energy so that they can have energy to work with, and then able to build new substances. So with a biochemical pathway, you have a starting point, whatever that might be, say glucose or sugar, and it's a pathway, so there are different steps in order to reach the end product. And with glycolysis, the end product is actually pyruvic acid. Pyruvate. That is the last step in the official process of glycolysis. Alright, there are different pathways and ways that we can categorize these different pathways. And the two major ones are catabolic and anabolic reactions. The catabolic pathways break down substances and release energy. And this would be, say, glucose. You break down glucose, or the process of glycolysis. You break down glucose, you end up with some energy, which is this energy released component, and you have your end product of pyruvic acid. Anabolic pathways, on the other hand, combine energies. They're going to use energy with different molecules to build new substances. You've probably heard of anabolic steroids. People might take those in order to build muscle. There's material in the steroids to help facilitate the growth or the building of those new muscles. So anabolic. All right, with catabolic pathways, if you forget how to keep the two separated, I always think about catabolic is catastrophic. And in terms of catastrophic, you're breaking things. All right, it may not actually be that catastrophic in nature, but with catabolic pathways, you're breaking things down, you're pulling things apart, and in that process of pulling apart, you release the energy. Amphibolic pathways have a dual role, and these can be a combination of the catabolic and anabolic pathways, break things down as well as build new substances. Alright, and this is figure 8.1. When you have these different reactions, they're usually combined. Alright, here you have catabolic pathways. You break this molecule down, and now you have simpler molecules, and in that process of breaking this one part away from the whole, you release energy, and then the anabolic pathways use energy, so the energy feeds into this reaction to combine these two subunits into the one complete unit. And that's what it says when they're coupled. Usually you don't have just a catabolic reaction or an anabolic reaction. All right, they occur maybe simultaneously, but they're in different steps, but you have different reactions going at the same time. And these molecules can move back and forth between different reactions. And so always in a cell, you have catabolic reactions going on while you also have anabolic reactions occurring. So why do you have these reactions? One is to break things down to get the smaller molecules to build new molecules, but it's also to create ATP. and ATP is your energy, the cell's energy. Now you think about money. Things are worth money. However, these items are not spendable until they're converted to cash. So ATP is the cash that the cells get when they break down nutrients. You pull glucose into your cells, and your cells break it down to convert it to the energy, the ATP. All right. Unlike money, it's hard for ATP to be stored. You just can't save it up. And so there always has to be a means of allowing your cells or bacteria cells to make more energy. Now ATP, we've already looked at this in terms of the structure of ATP. All right, ATP is adenosine triphosphate. You have your adenine group here, your sugar group, the ribose, and then you have the phosphate groups. If there's just one phosphate, then it's adenosine monophosphate or AMP. Two phosphates, adenosine diphosphate, and then the three full ATP molecules is ATP, adenosine triphosphate. Adenosine triphosphate is more energetic than adenosine diphosphate, and when you break this bond, then you release the cells. So ATP stores the energy in this molecule. You pull the phosphate group off, you release that energy that can go into some other reaction to fuel metabolism. Alright, so you've got this example here. Energy comes in from the form of catabolism, and that's that phosphate group. Phosphate gets bound to adenosine diphosphate really quickly, and now you have adenosine triphosphate. That goes to some chemical reaction that needs energy. Phosphate group is pulled off. That energy, this little star dude, is created that can go to fuel some reaction out here, and then you're left with adenosine diphosphate. And this process is constantly being done in bacteria cells as well as your cells. Alright, so what allows these reactions to occur? After you, well, assuming you have the base products, right, each step in a reaction is fueled through an enzyme. An enzyme is always a protein, and it is a catalyst to help chemical reactions occur under cellular conditions. And there is a video on just enzymes itself, but they are biological catalysts. They fuel reactions, allow reactions to occur faster with less energy. They're very effective in small amounts. You don't need like 100,000 copies of an enzyme. They act very specifically. They increase the rate of reaction. And of importance is they're not consumed or permanently changed, so they are recycled. And that's why you don't need, or that's why they are effective in small amounts, is because once the one protein does its thing with the energy or with the enzyme activity, it pops off and it can go right back into the process to repeat the reaction. Alright, so the catalyst only needed in a small amount. Alright, this is the enzyme. It increases the reaction rate, and as an enzyme, the catalyst is not consumed or permanently changed. Here are some examples of different enzymes, and if it's an enzyme, then it has the suffix ase. So just for example, hydrolase. Hydrolase is an enzyme that fuels the process of hydrolysis. Addition of water to break bonds. All right, a ligase joins two molecules. All right, that usually uses energy. And then you've got some examples here, but there's a lot of different types or groups of enzymes that are used in cells. But what you'll notice is they all end in ase to indicate the enzyme activity, and they all have specific reactions that they catalyze. Alright, here's a reaction of what's going on. The substrate is the molecule that an enzyme acts on, and here is the enzyme itself. And enzymes have an active site. Alright, the substrate is going to fit into the active site, and this is called an enzyme substrate complex. in that complex, the enzyme does its work, and you can start to see the change in the substrate. And then the products are released from the enzyme. Now you have your two separate molecules originating from this this one piece here. All right, one molecule broken down into two products. And then once this enzyme has released the product, it can go back up here and start all over again. All right, what this is called is the induced fit model. The enzymes are flexible and they're able to mold to the substrate, and they're able to complete the reaction because of that special fitting. Alright, enzyme-substrate complexes allow the chemical reactions to occur by properly positioning the reactants, the substrate, providing the activation energy required to start the chemical reaction, and then also stabilizing the transition state of the reaction. Right, really being a bridge between the starting point and the ending point. Alright, so here you have a figure that shows how energy is conserved. Alright, you have energy that is invested, and this is figure 8.5. Here you have the reactant, and if you have to break this molecule apart into the two parts down here, and you don't have an enzyme, it's going to take a lot of energy to get that done, and it's going to take a longer time. Alright, so more energy to begin with, longer time, and you get your products, and I guess that's okay. But what if you can do it with less investment, and you get the same result, less investment, and it's faster? Alright, that's a lot better. And so that is why enzymes are functional and healthy for all cells. Otherwise, all the chemical reactions really wouldn't be able to keep up with our cellular needs, and cellular metabolism would not occur. We would actually die. We would not be alive to even know that we died if enzymes were not available. Alright, sometimes enzymes can have cofactors. Alright, the cofactors come in and help alter the shape or allow the substrate to actually bind to the substrate, bind to the substrate to allow the products to be made. In this case, you don't have the cofactor, and even though the substrate might be able to bind to that active site, the substrate isn't formed. Alright, now how do you regulate enzyme activity? One is through temperature, and in this case you have an optimum temperature for the enzyme activity right at 40 degrees. At lower temperatures it might work alright, but it's not 100% effective, 100% efficient. On the other side, you actually have a shorter area, a shorter range in which the enzyme will work. Right? Because at higher temperatures, the higher temperatures are going to denature the proteins. They're going to break the bonds, and those proteins are going to fall apart, and without the proper shape, the protein is not going to work. The enzyme is not going to work. Alright, and that's what you have here. This is an active protein. You heat it up, and this protein then becomes denatured. It's inactive. Right? It doesn't have the proper form there. Alright, pH is also a factor. Right? We mentioned how pH is a growth factor for bacterial growth, looking at week 7 material. And this is why, because pH will affect enzyme activity. I also have your concentration of both the enzyme and the substrate. Alright, how much do you have of each will depend on how fast your reaction goes. Alright, and then the last bit about enzymes we're not going to get to in this part, but there are definitely control mechanisms in which you can help to control enzyme activity.